Well, we are going to start now. Welcome to the sounding board. My name is Nellie High Iredale. I'm the sounding board chair. This is Oceanids' 68th year, which is an official support group of the university. The sounding board is a joint effort of the faculty club and Oceanids. Let me the gallery view. There you go. Our honorary chair is Thespin Kapulakis, who's here with us, and she's the associate of the chancellor, and our president, Nigella Hilgert. In Oceanics, we raise money for grants and awards for students, support other UCSD events and activities such as the Student Grant Free Clinic. We offer many events for our members, and Sounding Board is one of these events. We would love to have you as a member, and I encourage you to join. It's only $35 per year. We are happy to see you now via Zoom. We have this event every first Thursday of the month throughout the academic year, except for January. Thank you so much, Judy Vaquier, for co-hosting. Now, I would like to introduce you to our speaker, Stephen Spector, MD. Dr. Spector is a distinguished professor of pediatrics in the Division of Infectious Diseases and director of the Mother, Child, Adolescent HIV program at UCSD. He has a long history of clinical trials and studies evaluating respiratory diseases, including influenza, parainfluenza, and respiratory syncytial virus. Dr. Spector has been involved with HIV/AIDS research since the beginning of the epidemic and has been involved in clinical trial groups for more than 10 years. Dr. Spector has served as a senior advisor to the William Clinton Foundation and to the President's Emergency Program for AIDS Relief. He has been honored for many of his contributions, including the prevention and treatment of HIV infection of children. Dr. Spector currently leads the phase 3 clinical trial at UCSD, evaluating the safety and efficacy of the Moderna vaccine. He will talk to us today about the COVID-19 vaccines, fact and fiction. Welcome, Dr. Spector. Thank you so much for being here with us. Would you like to share your screen? Perfect.
Thank you very much. And let's see if I can, there we go. Well, thank you. And it's good to see the faces and of some of my old friends and neighbors. And what I'd like to just go over is COVID-19 vaccines. I think that's been a very important issue of late, as everyone knows. And what I'd like to do is just to briefly give you an overview of what I will be discussing. First, I'll give you just a brief overview of SARS-CoV-2, the virus that causes COVID-19. We'll talk about the COVID-19 vaccine development. I'll review COVID-19 vaccines and those that have been approved for emergency use. I'll talk about the data supporting the efficacy of these vaccines. I'll talk about the activity of the vaccines against the variant strains, which have become very important as late. I'll also discuss whether or not people with previous COVID-19 can benefit from being immunized, as well as when will COVID-19 vaccines be available for children. And finally, I'll briefly touch upon the challenges in immunizing the US population against COVID-19. Well, these kinds of viruses that cause COVID-19, these coronaviruses have been around for quite some time. They're a large positive strand RNA virus, and they're very common cause of what we would call a common cold. On the other hand, over the last decade, we've come to appreciate that these particular viruses can cause very serious respiratory syndromes, including SARS, which was the original outbreak of SARS-CoV-1. It also is the cause of Middle East respiratory syndrome, and now SARS-CoV-2 as the cause of COVID-19. So it isn't as though these viruses have crept up on us, and what we had not appreciated until the past decade is, in fact, these viruses can cause extraordinarily serious respiratory conditions. Well, I think as everyone knows, the virus was first identified in Wuhan, China. Initially, it was thought that the outbreak was associated with a live animal meat market. However, since then, there has been no link particularly to that market, and it appears that person-to-person spread was going on considerably before December 2019, when it was initially identified. However, I think if you're following the news media, the origin of the virus remains a controversial topic, and I'm really not going to get into what I think the origins of the virus are. One thing I'd like to say that the likelihood that it came out of a laboratory is not particularly high. Again, as you know, transmission can occur through droplets, and that really is the predominant route in which we become infected with COVID-19. And I think that that really is an important point. And what we have learned, in fact, is that SARS-CoV-2, again, the virus that causes COVID-19, is extremely infectious. If you look, for example, just at San Diego and the implementation procedures that we have used as far as wearing masks and social distancing and good hand washing, it turns out that the cases of influenza are virtually nil. That's in comparison to what we have obviously seen with COVID-19. So what this tells us is how highly transmittable the respiratory droplets are that are carrying the virus that causes COVID-19. Much less transmission occurs through contaminated surfaces, but what we do know is that transmission can occur, and it can very efficiently occur between persons, even if a person is asymptomatically infected. Well, when COVID-19 was first identified, and people were looking for correlates of protection, they naturally looked at whether or not there was antibody being made by individuals who were infected with COVID-19. And as it turns out, it was clear that not only were people making antibody to the virus, but they were making neutralizing antibody. And that has been the guide-on for the development of COVID-19 vaccines. And what was interesting is people who were hospitalized, in fact, made higher amounts of antibody than those people who were not hospitalized and recovered from their infections without ever having serious illness. However, when those vaccines were being made, and even today, we do not specifically know what the immunologic correlates of protection are. We have assumed that neutralizing antibody is what is critical to controlling infection. That may in fact be the case, and that is the guide again in which our vaccines have been made, but we don't know, for example, if cell-mediated immunity is important for protection, or if in fact there are other antibodies that are critical in protecting us from getting serious infection. But with the information that was available, by June of 2020, there were more than 125 different COVID-19 vaccines that were being developed worldwide. And every kind of vaccine was being attempted. There were DNA vaccines, there were RNA vaccines, protein-based vaccines, viral vectors of different kinds, inactivated virus, and even in China, they were trying a live attenuated viral vaccine. So there were a huge number of vaccines being developed. Many of the vaccines targeted the spike protein. That was thought based on early immunologic data and also based on what was known with SARS-CoV-1 and MERS, that it appeared as though the critical attachment that the virus had to make to get into a cell was through the spike protein and ACE2 that is on the cells that are becoming infected. So there had to be a decision as to which vaccines were going to be prioritized. And it turned out that there were 7 vaccines that were prioritized in development. And these vaccines, for the most part, have been successful. There are clearly data on the first 3, each of which that have EUAs. I'll briefly talk about the AstraZeneca vaccine, which has not been put through the FDA in our country and is not an approved vaccine in the US. The Novavax vaccine, which I will not speak of, but I think we will be hearing data in the next month or 2 from the Novavax vaccine, which is a protein subunit vaccine against, again, the spike as well as the matrix protein. Sanofi is developing their vaccine. Their vaccine has been slowed down because the initial dose that they used was lower than appears to be necessary. One of the first things that I think is very important from any of the vaccines that we are discussing is that none of these vaccines can cause COVID-19. Just as if you have a tire or a steering wheel, you don't have a car, using fragments of SARS-CoV-2 is not going to give you a viral infection that causes COVID-19. So you do not have to worry about getting COVID-19 from any of the vaccines that have been approved within the US. But to test these vaccines, NIH set up what's called the COVID-19 Prevention Network. And unfortunately, this was given the title of Mobilizing the Force and Warp Speed. And I think that that was an unfortunate title. And the reason for that is that although the process of developing a vaccine was to a great extent accelerated for these vaccines, none of the critical steps were in fact skipped in vaccine development. So as an example, the research, which usually takes 2 to 4 years, and if it's accelerated, takes 6 months, took a much shorter period of time. Within a month, we had vaccines to COVID-19. And I'll explain to you why that is when I talk about the mRNA vaccines. Preclinical trials for vaccines normally would take 2 years, accelerated takes 6 months. In this case, the preclinical data, again, was greatly shortened. The clinical trials that very often will take 5 years and even in accelerated periods of time, 1.5 years, in this case, really took a period of less than 6 months. And approval, again, usually takes at least a year. Accelerated 6 months was done within a very short period of time. manufacturing and getting the vaccines out. So as you can tell, vaccines will frequently take as long as 8 to 15 years to be developed. However, in less than a year, we had vaccines that could be tested and approved for COVID-19. So I think it really is a great accomplishment of science that the vaccines were able to be developed in such a short period of time. And what really made this happen, and right out of the gate, the messenger RNA-based vaccines were clearly the ones that could be developed the most quickly. And these vaccines, for those of you who don't know, are based just on the principle that when you have DNA, it goes to messenger RNA, and the messenger RNA makes the proteins. And this is the basis for the messenger RNA. So these messenger RNAs, when placed into nanoparticles, in fact, when injected into muscle, will make proteins that are then recognized by the immune system. And this is a slide that shows the process for developing mRNA vaccines. The critical step, and the one that can take the longest period of time, is identification of the antigen that's going to be used and the amino acid sequence of that antigen. Again, fortunately for SARS-CoV-2, what could happen is that because it was already known that for SARS-CoV-1 and MERS, that the spike protein what appeared to be the most immunogenic protein, and there had been vaccines made against the spike protein for those other coronaviruses, the mRNA people, as well as those that were going to make recombinant vaccines, went after the spike protein. The amino acid sequence was identified quickly. And then, in fact, you found the RNA sequence that was going to make that protein. You can then take the DNA and put that in a plasmid, and that plasmid could make a lot of messenger RNAs. You then can purify that messenger RNA, put it into nanoparticles, and once you do the preclinical study, scale it up and go into humans. And this was done in an extraordinarily short period of time. So within a month of starting to make mRNA vaccines, they already had a vaccine that could be put into animals to look for toxicity as well as potential efficacy and antibody that is being made by these vaccines. So the first vaccine that was developed is the Pfizer mRNA vaccine. And this study enrolled 43,000 participants. The vaccine consists, I'm sure you know, if you got the Pfizer vaccine, of 2 doses, 21 days apart. And then they began to analyze their data 28 days after the first dose. And what they found with this vaccine is, in fact, that of the individuals who had symptomatic COVID-19, 8 had received the vaccine and 162 had received placebo. And when it came to severe COVID-19, only 1 of 10 had received the vaccine. So there was this vaccine efficacy of 95% for the mRNA Pfizer vaccine. So this was extraordinarily exciting and really indicated for the first time that mRNA vaccines could be used. mRNA vaccines have been around for the last 10 to 15 years, but there has never been a licensed vaccine using mRNA previously. And that's because these vaccines were going through that usual 8 to 15 year development process. And given that the mRNA vaccines are a relatively new approach, again, there was never an mRNA vaccine that had been licensed. This now shows the cumulative incidence curves for the Pfizer vaccine. And I think what is very clear, whether or not you look at the inset here or you look here, that the vast number of COVID-19 cases is in the placebo group, and very, very few cases are in the vaccine group. This now shows the adverse effects associated with the vaccine. And this is now looking at less than 55 years of age and 55 years old and older. And as you could see, the vaccines, and all these vaccines have pretty much the same kinds of adverse effects of some fever, fatigue, headache, chills, some vomiting, diarrhea, muscle aches, and some joint pains. However, there are very few what would be considered grade 3 or serious adverse effects, and very few, if any, grade 4. It subsequently has been found that anaphylaxis occurs occasionally in people, and it's estimated that it's somewhere between 1 in 2,000 and 1 in 1 million doses. The second vaccine that was approved with an EUA, so it's an emergency use, it's not fully approved, but it's an emergency use approval that these vaccines have, was the NIH Moderna study that again was an mRNA vaccine. And this again had a 1-to-1 randomization, and it examined the efficacy of prevention of symptomatic COVID-19. And this vaccine is given 28 days apart. Again, the Pfizer vaccine is 21 days apart. This is 28. And again, this is just what the spacing that the pharmaceutical companies decided to use. It isn't as though it's made in stone that you need to have the Pfizer vaccine exactly on 28 days. You can certainly have it after that. And same with the Moderna vaccine. You could have that vaccine a few days earlier, or you could have it somewhat later than 28 days, and you'll still get a booster effect with the vaccine. But these are the intervals in which the studies have been done. And this study was designed to enroll approximately 30,000 adults 18 years of age and over. And these are the efficacy data for the Moderna study. And that shows that of the 196 endpoints, 185 were in the placebo group, 11 in the vaccine group. And when they looked at severe COVID-19, 14 days after the second dose, none of the 30 severe cases of COVID-19 were in the vaccinated group. The efficacy was seen across all age groups, race, ethnic backgrounds, and gender. So again, between the two mRNA vaccines, you have a 94 to 95% vaccine efficacy. Extraordinarily exciting, and frankly, much better than any of the investigators ever thought that a vaccine would be against COVID-19. So this again is an extraordinary success of science and moving the vaccines forward. And this now shows the cumulative incidence curves. And once again, clearly, almost all the endpoints are in the unvaccinated group. The Moderna vaccine has very similar side effects as the Pfizer vaccine. Again, there's fever, headache, fatigue, joint pains, nausea, vomiting, chills. Very few grade 4. These are grade 3, grade 2, and grade 1. And but you do have, and what's interesting both for the Pfizer as well as Moderna's vaccine is that those under 65, and particularly those under 50, are more likely to have more of the adverse effects than are older people. Now, the final vaccine that has been licensed so far that has the EUA is the Janssen vaccine, the J&J vaccine, and this is a slightly different vaccine. This is a vaccine that uses a vector that's from adenovirus 26, which is a human adenovirus. And that vector is not replication competent. It can only do a single cycle. And what that does is it encodes for the spike protein. And so this vector replicates one time, makes the spike protein, and the immune response is to the spike protein. Now, this vaccine, or at least the vector, has had considerable history, and it's been used for a number of vaccines. And at the time when J&J Janssen was developing their vaccine, they had already immunized greater than 90,000 people with this vector. So they felt quite comfortable that this vector was going to be safe, and that all they needed to do was to put their spike protein into the vaccine. And once they had the right configuration of their protein, they would be able to produce their protein safely with their adenovirus 26 vector. So this was another study that was a 1-to-1 randomized controlled study. They enrolled over 43,000 participants that received a vaccine. And the advantage of this vaccine is that it was only requiring a single dose of vaccine. Now, whether or not at some point they'll be required a booster for the Janssen vaccine, just as with the mRNA vaccines, we might require a booster for Johnson's vaccine, and we may in fact require a booster for the mRNA vaccines. But these are now the data for the Janssen vaccine. There are a couple of important things that need to be remembered about this study. The first is it occurred a few months after the Moderna and Pfizer vaccines. So the amount of circulating COVID-19 was considerably greater when the Janssen study was going on than when the other studies were going on. In addition to that, they studied the vaccine in the US, Brazil, and South Africa. Again, it's a single dose vaccine. Here you see here the point estimates for the US, in which moderate to severe COVID-19 at 14 and 28 days was 76% and 72% efficacy. And for severe illness, it was approximated 72% and 75%. What they also found is in Brazil, as well as in South Africa, they also saw similar efficacy. This is particularly important in South Africa, and I'll show you some other data that demonstrate that the Janssen vaccine, in fact, is very good in South Africa. This now shows those incidence curves that I've shown you for the other two vaccines. And what you can see here is that although the overall vaccine efficacy post 28 and 60, post day 28 was 66%, that it's interesting that there seems to be a flattening of the curves. And when you look at the data, what turns out to be very important about the J&J vaccine is that beyond 49 days, when you look at severe COVID-19, there are virtually no cases of participants who receive placebo who develop severe COVID-19, as opposed to those that did not receive the vaccine. So we can see here that, in fact, the vaccine post 49 days was very effective in preventing severe disease, which is really what we want to do with our vaccines. The adverse events, again, are very similar. I'm not going to go through all these again, but they're all very similar: fatigue, headache, myalgias, nausea, fever. And again, very few grade 3 toxicities. The final vaccine, and this vaccine does not have an EUA, and the data are controversial, but this is the most up-to-date data that I could frankly get off of the news media, because AstraZeneca has not presented yet the data for their US study, but they did enroll over 32,000 participants. They were randomized 2 to 1, two to receive vaccine, 1 placebo. They've had 190 symptomatic cases, and they have an efficacy of about 75%. They have an efficacy, they have said, of 85% in adults older than 65 years of age, and 100% efficacy in preventing severe or critical illness. So these are all the data that we have so far on the US study. It's anticipated within the next few weeks that AstraZeneca will present their data to the FDA. They also use a vector, and it's an adenovirus from a chimpanzee, so it doesn't replicate in humans. And again, it'll be very interesting to see what happens when this vaccine goes to the FDA. So there are a number of questions that I'm constantly being asked, and I thought I would try to answer some of them, because I'm sure that these are some of the questions that you have as well. One is, do the current COVID-19 vaccines prevent infection in the real world? And another is a single dose of one of these mRNA vaccines effective in preventing COVID-19? So first, what I'd like to do is show you a study that's out of Israel. And they actually did a very interesting study. They knew that those people who are over 70 years of age are the ones who are getting the most severe illness, required mechanical ventilation and hospitalization, and were doing the worst. And they knew that people less than 50 years of age were at lower risk. And they looked at what was going on between people who were 70 years of age and over, and 50 years of age and under. And initially, they had a ratio of 7 to 1 of older people to the younger group. Once they had strict national stay-at-home orders that brought that down, but then they began to immunize. And they were only immunizing those people who were 70 years of age and older. And within a relatively short period of time, within a month, they had dropped that ratio from 7-to-1 to less than 2-to-1. So that was the first real indication on a national basis that, in fact, the vaccines seem to have an impact on the real world and cases and severe cases of COVID-19. There was another interesting study that was done out of UT Southwestern, and they began to immunize their entire workforce at the medical center. And they didn't have enough vaccines, so it took over time, just as we had that problem of immunizing everybody. And so they looked at those people who had not been immunized, at those people who were partially immunized, and those people who were fully vaccinated. And as you can see here, when you look at the percent of people who got COVID-19, those that had not been immunized were 2.6%, partially immunized 1.8%, and fully immunized 0.05%. So you can see here that just on this basis, they in fact could show that immunizing the employees that are part of their health center could in fact prevent people from getting COVID-19. The next question is, can one dose of the Pfizer or the Moderna vaccine prevent infection? And these data are from the CDC, and they looked at 3,950 participants. The bottom line is that one dose did provide some protection after 14 days. And in fact, what they found is the efficacy of the first dose, just getting one dose, was about 80%. Now, the efficacy of 2 doses we've seen is 90% or over. So there is an advantage to getting that second dose. The other thing that we do not know is the duration of protection. So one might think that those who received a booster are going to have higher antibody and they're going to have a more durable immune response than those who got a single dose of vaccine. So that is why it is recommended that people still get the 2 doses, although again, a single dose does afford some protection. What period of time that protection lasts is unknown. So another important question that is coming up more frequently is, do these current COVID-19 vaccines protect against evolving variant strains? And first I'll tell you the bad news, and then I'll show you some better news. And that is that the AstraZeneca vaccine was tested in South Africa in approximately 2,000 people. And what they found is against the B.1.351 variant, which is the South African variant, that the vaccine did not work. In fact, it provided virtually no protection against the development of COVID-19. And they not only stopped their trial, but they've stopped immunizing people with the AstraZeneca vaccine because it clearly did not work against this variant of the B.1.351 strain, the South African variant. Now, the Janssen vaccine, J&J, was studied in different countries. It was studied again, as we heard earlier, in the US, Brazil, and South Africa. And when you look at the South African variant, and again, these were predominantly the B.1.351 variant, the vaccine efficacy preventing against severe or critical illness was 82%. So it appears as though the Janssen vaccine does prevent illness with the South African strain, It also appears to provide almost 70% protection against the Brazilian variant. And again, in the US, it had an 86% vaccine efficacy for severe and critical illness, and 100% after day 49 of having received the vaccine. Now, how about the mRNA vaccines? Well, as of now, we do not have clinical data in South Africa or Brazil, but here is a study looking, I'm looking at Moderna and looking at the activity against the UK variant and looking at neutralizing area antibody against the B117, which was first identified in the UK and is rapidly becoming the dominant strain in the US because it replicates so much more efficiently. And the neutralizing titers were basically no different between what I'll call the wild type D614G virus that the vaccines were all made against and the B117 variant or the UK variant. In addition, although there is a decrease in the neutralizing ability of the vaccine against the B1351 variant, again, the South African variant, it still neutralizes this virus. However, it neutralizes it at about at least twofold less than what has been seen for, again, the wild type strain. So in fact, there is evidence that there is some protection, but the question is, can you in fact give a booster with a variant vaccine that on top of the vaccine that you've received now? So Moderna rapidly has developed a vaccine against the South African variant, the B1351, and they are now testing this vaccine to see if, in fact, if people who have received previous doses of the original vaccine, if they will get a booster and have higher protection against the B1351 variant. And those studies are going on now at the NIH, and it is thought that within a relatively short period of time, we will know if we require a booster against 351, or if in fact this newer vaccine will be used as a booster. So they are going to be comparing a booster using their original vaccine versus now their variant vaccine to see what the activity is against these variant strains. So should people with previous COVID-19 be immunized? And here are some data. The bottom line is yes, because one, it's known that people can be reinfected. But I think what it also shows you is that if you look at the antibody titers of those people who had infection with COVID-19 and were symptomatic, and then if they received a vaccine, they clearly have higher neutralizing antibody than they had prior to their being immunized. So I think that this is an important point, and one in which it also appears as though people who have been previously infected make higher antibody than those people who have not been previously infected. So one can make a very good case, at least based on antibody response and based on the fact that we know that people who have had COVID-19 can have reinfection. For people who have been immunized or have had natural COVID-19, that being immunized potentially can improve protection. Finally, I just want to speak very quickly about children and COVID-19. What is known is that children can have similar symptoms as adults. However, their infection is usually mild. Many of the cases are asymptomatic or of mild infections. But clearly, we need to immunize children for a number of reasons. One are the obvious ones. We want children back in school. We want to protect their teachers. We want to get them back in athletics and their other activities. We want to improve the socialization that's so important for children. But there is even a more practical reason, and that is that almost a quarter of our population in the US are children who are less than 18 years of age. So there are almost 50 million children who are less than 12 years of age. So we really need to have a vaccine for children who are less than 18 so that we can protect our population if we're going to achieve herd immunity, which is going to require at least 70 to 80% of our population being protected of our population of 331 million people. So Pfizer and Moderna have expanded their studies, and they're going to be studying children who are between 12 and less than 18. Pfizer actually has had their vaccine approved for children 16 and 17, but they're studying it in those 12 to less than 16 years. And Pfizer just yesterday released data on their study of children between 12 and 16. And they found, again, of over 2,000 children that they enrolled into this study. Of their COVID-19 cases, none occurred in the vaccine group, 18 in the unvaccinated placebo group. So clearly we're seeing the same efficacy in 12, 16-year-olds as was seen in the adult population. So there are studies that are going to be going on in children that are less than 12 years of age. We, in fact, are going to be starting a study very soon of the Moderna vaccine. This vaccine is going to be examining children who are between 6 years to less than 12 years, 2 years to less than 6 years, and 6 months to less than 2 years. Once the dose is identified for this group, there then will be a 6,000 child study, and there'll be randomization of 3-to-1 vaccine to placebo. But everyone eventually will get the vaccine in relatively short order. The adult dose is the 100 microgram dose. And one of the questions is can you get by with a lower dose in children? And in fact, there's some question whether or not, even in adults, whether or not you need the 100 microgram dose, because there are now compelling data that show that 15 microgram dosages actually appear to generate the same neutralizing antibody as the 100 microgram dose in adults. So when are vaccines going to be available for children? Well, I anticipate that by the summer, we'll have vaccines for adolescents who are 12 to less than 18 years of age. My hope is that by the fall, there'll be vaccines for children between 6 and less than 12. For the younger children, it's going to take a longer period of time. For those that are 2 years to less than 6 years, I anticipate by the end of the year, we'll have those. And for those in the younger group, my expectation is we'll be into the first quarter of 2022 before we have a licensed vaccine for children who are 6 months to less than 2 years. Finally, there are many challenges to immunizing our population. Again, we want to achieve at least 70 to 80% of the population being immunized. We need to have one message for the entire country. This has been a challenge until recently. We need to get the vaccines out, and we need the public to be accepting of the vaccines, which honestly, I think is really increasing substantially. And finally, we need to get to the populations that historically have been underserved. So we need to make sure that minorities, people of color are being immunized because they have been disproportionately impacted by COVID-19, and they disproportionately have underlying conditions that make them at even greater risk for having serious and life-threatening COVID-19. So I'll stop here. We're all in this together. It's been a long haul in getting these vaccines going and approved, but I think we are there. And hopefully as many of you have been immunized as possible. And I just want to show you, just to do our study here at UCSD, in which we enrolled 335 people. This is the group of people that were required for us to accomplish the vaccine. So it really took an enormous effort to get that vaccine study going, and this happened all across the country. So thank you, and I'll stop sharing my screen, and I'd be glad to answer questions.
Thank you so much, Dr. Spector, for this fabulous presentation. We have either the chat that we can ask the questions there, or we can raise our hand either digitally, or maybe if I can see you raising your hand, I can, Liz. Yes, please, Liz, go ahead. Would you like to unmute yourself? Go ahead.
Yes, I have grandchildren, and I'm irritated that there's such a delay in getting them immunized, especially since they announced today that they have one that's good for essentially preteens, and they think they'll be able to go back to school in the fall. If they've got it, why not immunize them now so they can go to summer camp? That's what I want to know.
Well, again, the data just came out yesterday.
Yes.
They'll have to, finalize what their data are, present it to the FDA, and my expectation is within the next, I would say, given the process and the data within the next 6 weeks, you probably will have the Pfizer vaccine receive an EUA from the FDA so that those between 12 and 18 will be able to be immunized.
Why not tomorrow?
Well, there is a process, and the data need to be reviewed. We all again, on the one hand, because of the situation that we've all been put in, every day seems to be too long. On the other hand, the development of these vaccines and the EUA and actually getting it into people is just so unprecedented that you really have to say that there really has been a tremendous job in moving these vaccines forward. So I understand your frustration. I think many of us on this call have grandchildren who we'd like to see immunized. And I think we will get there.
Good.
Thank you, Liz. We have a question on the chat room. It says, is the second vaccine stronger than the first or different somehow? Pfizer and Moderna.
Those two vaccines are very similar. The mRNA vaccines, the efficacy is the side effects. I mean, they're almost duplicates of each other. So I would say they're as close to the efficacy and everything else that you could possibly have on the two vaccines. And I think it's really great that that was demonstrated, because when Pfizer came out with 95% efficacy, it it was almost too good to be true, particularly since that was the first mRNA vaccine that was going to potentially even get an EUA. So when the second vaccine came out and that was identical, it really gave additional proof that these vaccines work.
Thank you, Dr. Spector. One more question from Josephine. My family members who are teachers in Canada are getting the AstraZeneca vaccine. Why was it approved for wide distribution in Canada and not the US?
Well, there are a number of things. The AstraZeneca vaccine has had a number of challenges since they started to release data. Having said that, it looks as though the vaccine does have efficacy. It may not be as good as some of the other vaccines, but appears to prevent serious infections. The complications associated with it have been a concern. So you may have heard that the EU is now saying that they're only going to immunize people who are over 60 years of age because the thromboembolic phenomenon appear to be in people who are less than 60, more in women than in men. But again, there were 14 cases. And I think at this point, AstraZeneca has completed their US study. They're getting their data together and they'll have to present it to the FDA and the FDA panel, and they'll have to review all the data. It's been a very controversial vaccine. My expectation is that it will at some point be approved in the US, but it hasn't been because no data have been presented to the FDA, and the FDA is waiting for the US study data.
Thank you. And then from Judy, is Moderna or Pfizer still following the patients in the trial to see how long the immunity lasts?
So we are following all the participants in the study. And so we have, of our 300, as an example here at UCSD, of the 335 that we initially enrolled, over 300 are still being followed. There were some who were immediately offered the vaccine who got it, who then dropped out of the study.
And one more question. If you have had the 2 Pfizer shots and the Moderna booster is shown to be effective against the South African variant, can we get that booster?
There are no data. We'll have to wait and see. My expectation is that any one of these vaccines would boost the other, but you really would have to wait and see what the data look like. And I think that would come relatively quickly.
Thank you, Dr. Spector. One more question. Is an mRNA vaccine potentially more dangerous for children, especially young children? It makes me think of DNA, but I don't know if that only applies to the virus rather than the patient.
Well, again, this is mRNA. It makes its protein and then is rapidly degraded. It does not integrate into cells. So I would think that there is no risk to children or adults.
Thank you very much. And we also have from Teresa, please make this talk available so we can share it with our friends and family members. Yes, we are recording. And thank you for giving us this talk from all of us. Let me check if we have any more questions. These are the ones that I have in the chat. If anybody has any other question that I'm missing, if you would please like to unmute yourself. Am I missing anybody? Are we okay? Mary, did you have a question?
Oh, I'm sorry. No, I just wanted to just say that it was a great talk and thank you. That's all.
We all, well, then, Dr. Spector, thank you very much for this amazing talk. We all had a lot of questions.
Thank you. Bye now.
We are all muted, but everybody is clapping. So thank you so much. And we will see you next time, the first Thursday of next month. We will have Professor Chris Wills. Thank you all very much. Bye-bye.